Hello, everybody. Good evening, everyone. This is Tithi here. How are you all doing? Can you people hear me? Please drop a chat message. Please drop a message in the chat if you are able to hear me. Good evening, Sakina. Good evening, Biotechy. Welcome to the live session, guys. Good evening, Shraddha, Shipra. I'm doing good. Thank you so much, Biotechy. How are you doing? Hello, Rajeshwari. Good evening, Rosie. It's good to see you all. So many people have joined. Good to see you all here. Yes. So as you already know that I'm going to solve a genetic mystery. Not me, we together are going to solve a genetic mystery today, right? So what we are going to do here is we are going to solve some questions, some part C, some type questions from the subtopic of Mendelian genetics. Okay. Now, Mendelian genetics is a very important segment for CSIR exams from the CSIR syllabus. Mendelian genetics is very important. Like it is one of the important topics, and it is not only important for the exam point of view, it is also important for your basic understanding of the entire inheritance biology, right? So are going to take up some questions which has come in previous year question papers, and also I have added some extra questions, okay, which can very well qualify as part C question, okay, and then we are going to solve it all together. Okay, so if you guys are not ready with your pen and paper, please get it. All right, because a lot of writing you will have to do, because most of the questions from Mendelian genetics you cannot solve it on just like that. You write it down, isn't it? It's like kind of it will be like a mathematics class, so you cannot solve a mathematics just mind. You have to write it down. Okay, so we are going to do that in today's class. So please be ready with your pen and paper. Okay, so are you people ready? Neetu, Sakshi, Ayushi, Janvi. Okay, biotech, so you already know. All right. Thank you so much. I'm glad that you are here. Okay. Yes. So we are going to solve some questions. Don't worry. I have picked up some tricky questions. Okay. All of you ready? Okay. Can I have some BIOs in the chat so that people are ready and we can start? All right. Let's get started then. Okay. So this is one of the questions which says, which of the following representations of chromosomal arrangements in a meiotic metaphase 1 best explains the law of independent? Okay. See, I just got the information from our team that the video is breaking. Okay, so what I will do is I'll get video so that we can have an uninterrupted streaming here. Okay, is my audio better now? Because I have turned off the video. Okay, great. Okay, so let's continue. So, which of the four representations of chromosomal arrangement in the meiotic metaphase 1? Okay, now, what about this? What about the independent assortment? So, independent assortment, what does it say? It says that if two genes are present differently on different chromosomes, okay, all the possible combinations can be there in the gamete at the equal probability, right? So independent assortment says that the genes which are present on different chromosomes can come together in any possible combination. There are four options given. Okay, so now this is during metaphase 1. This is the arrangement which is shown for metaphase 1. Okay, so you have to identify which explains the law of independent assortment correctly. Can we have some results here? Can we have some answers here, please? See, for solving this question, you need to have a understanding of the cell cycle. Okay, just genetics will not help you here. You have to understand the idea of cell cycle as well. Okay, so you should know that you have to know genetics or the Mendelian principle. Then only you will be able to understand and answer this question. Okay, it's my okay, it's still breaking. Is the voice better now? See, so far, Sakina, Shraddha, Biotic, I'm so sorry your answers are wrong. See, all three of you have given the answer option 3. Okay. Now, option 3, what it is showing, that what is this line meaning? This line means that this is one chromosome, right? And these two means they are joined here, means they are the they are sister chromatids, right? They are the sister chromatids. So sister chromatids are formed after the result of the one chromosome, right? So if you see 3, so in 3, A and B are present on the same chromosome. Okay. So if A and B are present on the same chromosome, then they will be linked, right? The genes present on the same chromosome have a tendency of getting inherited together. Is it not? Right? So how can 3 be? Because we are talking about dependent assortment. Linkage was a phenomena that was never encountered by Mendel. The 7 characters that Mendel studied, they were present on 7 different chromosomes. So that genes which are different genes will get assorted differently. Okay. So option 3 cannot be possible because it is the genes are linked here. So here the answer will be option 1. Why option? Option 1, what is it happening? That these two are coming together and these two are coming together. A, capital B are coming together. Small b are coming together. Okay, and they are also present in different chromosomes. In this part, A is together with small b and small a is coming together with capital B. Okay, so this is how, okay, its audio is not clear. All right, let me switch my internet. Okay, I'm very sorry for this disturbance, everyone. Please stay back. Please do not leave the class. I'm going to change the internet and let's see. All right. How about now, guys? Is it better? Hello, everyone. I have connected again. Please let me know, is the voice better? Okay, excellent. All right. So now let's continue. Okay. And yes, guys, so I forgot to tell you one more thing. Everybody stay till the end. Okay. At the end, you will get something free, completely free from Biotechnica that will definitely help you in preparing for your CSIR exam. Okay. And at the end, I will also give you the link for the Zoom class where we can have a one-on-one kind of doubt solving session as well. Okay. Yes, I will re-explain this entire question once again. Okay. Right. So in this question, it is talking about independent assortment. Okay. So very few of you answered, but you all answered 3. Now 3 cannot be possible. Why not? Because in 3, A and B are shown on the same chromosome, right? A and B are present on the same chromosome. So the genes which are present on same chromosome, what we call them? Linked genes, right? Is linkage something that was observed by Mendel? Did Mendel encounter linkage? No, right? All right. Yeah. So the Mendel, he never encountered linked genes. Okay, he never encountered linkage. All right. So Mendel gave the independent assortment because that is what he understood from his experiments when the genes are present in different chromosomes. Okay. So independent assortment is something which is followed by genes on different chromosomes. Okay. So that's why option 3 is not at all possible. Okay. All right. Now, what about the other options? Okay, so here the answer will be option 1. Why option 1? Because this is the arrangement which is shown in metaphase 1. After metaphase, the meiosis 2 will also take place where the sister chromatids will divide. Right now, in this scenario, these two are coming together. That is capital A, capital B coming together. Here, small a, small b coming together. Right. In this situation, it is capital A, small b coming together, and here small a, capital B coming together. Okay. So these are the 4 different uh, genotypes in the gametes which is possible, and each possible with equal probability. Okay, where this option 4 is also showing the same arrangement. Okay, but it is saying either this or this, and that is not possible. Like, that is not according to what independent assortment said. Independent assortment said that all the possible combinations are can be there in the dihybrid crosses with equal probability. Is it followed, everyone? Okay, so in this question, answer will be option 1. Right. So if you had the knowledge of cell cycle, if you have the correlation of how to link Mendel's principle with the movement of chromosomes in the during the cell division, okay, then you will be able to answer it very clearly. Okay. Yes. What is shown in option 2, Shraddha? Yeah. So, Shraddha, what is option 2 showing is, I'll draw it here. It is saying that this is one sister chromatid. So, it is saying one chromatid has capital A, one will have small a. Okay. Now, that is absolutely not possible, right? Because sister chromatids, they will have the exact same alleles present on them, but because the sister chromatids are formed by the DNA replication. Do you get me, Shraddha? All right. Okay, so let's move on to next question. Right. Now, in the Mendelian genetics, the questions which are related to Mendelian genetics that are asked in CSIR exam, so they will be majorly related to the probability part. Okay, so here the probability calculations you have to do. Right. Now, I have often seen students who run away from genetics or run away from biology because of the fear of maths. Okay, so here it's not the mathematical calculation that is not difficult. Okay, so you will see that the mathematical calculations are very, very easy. What will be difficult is how you apply the concept of genetics in the mathematical problem. Okay, so let's see this question here. Gene A, B, and C control three phenotypes which assort independently. A plant with the genotype this is selfed. What is the probability for the progeny for at least one of the phenotypes controlled by the genes capital A, capital B, and capital C? Okay. So what is given here? Everybody, I want you to write down the cross. So cross is between which genotype? Cross is between here, capital A, small a, capital B, small b, capital C, small c. This has to be selfed. Okay. What is the probability for the progeny at least one of the phenotypes is controlled by capital A, capital B, capital C? Okay, so basically it is asking about the probability of this kind of progeny, okay, where it is capital A, this kind, or even this genotype, okay, or even this genotype, okay, that is at least one dominant character is present. Now, when it is at least one dominant character, means there is no upper limit. There can be two dominant character present, there can be three dominant, all three dominant character present, etc. Are you people getting my getting it? What is asked in the question? Okay, so if we have to find out the probability of all the progenies with one or more dominant character present, in that scenario we have to look for all the different possible combinations, right, and then find their individual probability. So that will be a long task, and definitely you don't have that much time in CSIR exam to do it. So what we can do here, we can do a shortcut. What will be the shortcut? See, the ultimate probability will be equal to 1. Sakina, you are absolutely correct. Biotechy, I'm sorry, your answer is wrong. So this ultimate probability will be equal to 1. The probability of, 1 second, I'll just make it better. Okay. So probability of all recessive phenotypes, okay, plus probability of at least one dominant phenotype will be equal to 1, right? So at least one dominant. So what is the lower limit? Lower limit is at least one has to be dominant. Okay, so what class is not included here? The class where all the phenotypes are recessive. Don't draw the table. Don't draw the table, Shraddha. It will be waste of time. Do not draw it. Okay, we'll do the probability. Okay. Yes, Lavanya, your answer is also correct. Very good. Right. So if you have to, what we have to calculate this probability of at least one dominant. Okay. So probability of at least one dominant will be equal to 1 minus probability of all recessive. Right. Now, once you have got it, now this probability is easy to calculate. Why? Because all recessive will have only one possible genotype. Can you guys quickly write it down? What is the possible genotype of all recessive? Yes, anyone? What will be the genotype of all recessive? All recessive can have, absolutely correct, Lavanya, sorry. So what will be the genotype? Genotype of all recessive will be small a, small b, small c. Right? Absolutely right. So what you have to do, you have to find out the probability of this genotype in this particular cross. Okay. Now how to do that? So you write down the cross here. Now it is clearly mentioned that the genes are independently assorting. Okay. So we will separate the genes. Fine. So probability of this genotype is basically equal to probability of 3 independent events occurring together. What is it? That small a come together. The second event is small b come together. The third event is small c come together. So their combined probability will be equal to multiplication of their individual probability. Okay. So now just for separate the genes. Okay. So for gene A, what it will be, the cross is this. So you can make a cross quickly, monohybrid cross, if it is not in your fingertip. So what is the probability of small a? It will be equal to, you can make up monohybrid cross quickly and you will know that it is 1 by 4. But by now it should be on your fingertips. Okay, if you are revising for your CSIR this term examination, then it should be in your fingertip that it will be 1 by 4. Similarly for gene B, the cross is this. So probability of small b is again 1 by 4. For C, again 1 by 4. Right? Now, if you combine them all, so probability of all recessive genotypes is will be equal to 1 by 4. 1 by 4 multiplied by 1 by 4, that is 1 by 64. Okay. Now, this is not something that was asked. What was asked was at least one dominant. Okay, so that will be equal to 1 minus this value. So 1 minus 1 by 64, it will give you 63 by 64. Did you all understand? What do you think, guys? Is it not easy? So math was not, definitely was not the hard part, right? How you apply the concept of genetics, that was the tricky part. So the answer will be option C. Is it understood, everyone? Everybody, can I have some quick responses? Did you all understand? Then we'll go ahead and go to the next question. So by the way, if you guys have any doubt, any question here, okay, please write it down. So in the end of this session, in end of the live session, when we have this Zoom class, okay, so you can join the Zoom link and you can ask your personal queries, okay. Moving on to next question. Now, this is also a previous year CSIR question. So assuming that A, B, C, and D genes are not linked, the probability of a progeny being this, the genotype is given from a cross between these two parents will be. So again, as we did in the previous question, this question is pretty easy. Okay, in fact, it was a part B question, but I included it here so that you guys get an opportunity to apply the knowledge that you got in the previous question. So here the cross is again same, that is the genotype of the parents are given. So you just apply it here. All right. And then you find out the probability of this genotype. Okay. Now, I expect that you people will not go for making the Punnett square. Punnett square is a good and scientific way, but it is a tedious way. You cannot do it for more than dihybrid crosses. Okay, so that will lose a lot of time. अगर आप पुनेट स्क्वायर बनाने जाएंगे तो बहुत सारा टाइम चला जाएगा और हो सकता है कि आपको आंसर सही भी ना मिले. तो इसलिए we will do it with the help of probability. Okay, so in the probability, what we will do is, again, we will separate the genes, right? So what is the genotype we need here? So probability of this genotype we need. I want all of you to solve on your own as well. I'm solving it here, but try to solve it on your own as well. So this will be given as probability of capital A small a multiplied with probability of capital B small b multiplied with probability of small c multiplied with probability of capital D small d. Right? So आप एक एक करके जीन यहां से अलग कर लो. So जीन A के लिए क्या हो जाएगा? Probability of capital A small a will become equal to how much? No, Sakina, this time you are wrong. So cross here, he is capital A cross with small a, small a. Okay. The next generation, entire progeny will be capital A, small a. So in that scenario, what will be the probability here? Probability will become 1. 100% of the next generation will be a heterozygote. Similarly for B, we want what? Capital B, small b. Okay, so the cross is between capital B, small b and capital B, capital B. So in this scenario, the probability of capital B will be half, right? Half of the population will be homozygous dominant, other half will be heterozygote. Yes, biotech Kavya, excellent, you are correct. Okay, now for C again. Okay, so again it is small c with small c. Definitely the entire progeny will be small c. So probability is 1. For D, if we have to calculate, the cross is capital D small d with capital D. So again, half of the progeny will be heterozygote, other half will be homozygous dominant. Okay, so you substitute the values. So the probability will come as probability of this genotype in the next generation will come as 1/4. ये सारे values multiply हो जाएंगे. So 1/2 multiplied by 1/2 will be 1/4. Answer is option C. Okay, understood, everybody? Okay. Now, another question I have taken, and this question was in the last CSIR exam only. Okay, this question was asked in the last CSIR exam. So, in Drosophila, balancer chromosomes are used to keep all the alleles on one chromosome together. A balancer contains multiple inversions so that when it recombines with the corresponding wild type chromosome, no viable crossover products are formed. Balancers also carry an allele for the dominant phenotype. So balancers contain a dominant phenotype. Okay. Now it is mentioned. A Drosophila male with sepia eye color is crossed to a female carrying a third chromosome balancer. The allele for sepia phenotype is present on chromosome number 3 and is recessive for the wild type eye color. Okay, so what is mentioned so far? That this sepia, okay, the the sepia allele is present, and that sepia allele is present on chromosome number 3, and it is the recessive allele. Okay, recessive to the wild type. The dominant marker for TM6B, which is the balancer chromosome, is the tubby phenotype. Okay, means tubby phenotype will be the dominant phenotype. Tubby phenotype is dominant. Okay, so if we are saying SE is the sepia, okay, so let's write capital S for this tubby phenotype, okay, so that it becomes easy for us to understand. So capital S I'm writing for dominant. Okay, so this forms the sepia eye and this forms the tubby eye. Okay, further, an individual homozygous for TM6B balancer does not survive. Now, F1 progeny with tubby phenotype is sibmated. Now, the meaning of the word sibmated is it is crossed with an individual of the same genotype. Okay. The F2 progeny is expected to have which of the combination here, or which of the phenotype and in which ratio. Okay. Right. Now, what we will do is we will do the cross now. Okay. Now, see, the male is the dominant, the Drosophila male was taken, was having sepia eye color. So, sepia is a recessive. That means the male here is homozygous recessive. Okay. So, it is SE, SE. Who is that? It is the male. Okay. Now, he is crossed with a female carrying the balancer chromosome. And on the balancer chromosome, the dominant allele is present, which is forming the tubby phenotype. Okay. So, this is crossed with the female who have the balancer chromosome. That is, it has the S allele. Okay. Now, one allele for sure is S. Okay. The other allele cannot be S here. Why other allele cannot be S? Because it is clearly mentioned that homozygous here will not survive. Homozygous for balancers will not survive. Do you get me? Does not survive. Okay. So it is heterozygous, means other one has to be SE. Fine. Now the F1 generation, what you will get? Okay. So in the F1 generation, you will get either SSE, okay, or SESE. Okay. Now the F1 that we took, it is mentioned the F1 progeny with the tubby phenotype is submitted. Means we have taken only this individual and we have crossed it with another individual of the same genotype. Kaushik, I'm sorry, the answer is not that. Afifa Nazri, yes, you are correct. Good. So what we have here, so now we do the cross. Okay, so we will do the cross here. So F1, which F1 we have taken? The one with the tubby eye. Okay, so tubby eye will have which genotype? SSE. Okay, so SSE crossed with another capital SSE. Okay, now make the Punnett square and see. So S and SE come here, S and SE come here. Okay, so capital S, capital S, capital S, SE. Capital S, SE, here SE, SE. Okay, now what does it say? That two balancer chromosomes, any individual who is homozygous for balancer chromosome will not survive. That means this part will not survive. Okay, so in the F2 generation, what we will see? We will find this part and this part. So this genotype will form sepia eye and this genotype will form the tubby eye. Okay, so it will be tubby is to sepia will be how much? 2 parts of tubby is to 1 part of sepia. Okay, or if you reverse it, sepia is to tubby will be 1 is to 2. So here answer will be option 3. Sepia and tubby flies in the ratio of 1 is to 2. Okay. Right. Now moving on to one more question related to the probability and Mendelian genetics. The fur color in rabbits is determined by a pair of alleles, capital B, small b. Capital B, and capital B, small b are black in color, whereas small b are white in color. Okay. Now the farmer wants to breed the rabbits for sale. True breeding rabbits, true breeding white rabbit females, okay, they breed poorly. The farmer purchases a pair of black rabbits and this mate and produce 6 black and 2 white offsprings. The farmer immediately sells his white rabbits. If he performed random crosses between the pairs of F1 rabbits, what proportion of F2 progeny will be white? Now, let me break down the question to you all. So here we are talking about fur color of rabbits. Okay. So rabbits, the fur color is controlled by gene B, which has 2 alleles, capital B and small b. Okay. So capital B is dominant and it codes for black color. Small b is recessive and it codes for white color. Okay. Now definitely when the farmer wants to breed the rabbits for sale, so white rabbits are more attractive, right? So they sell better. So the easiest way of getting white rabbits will be what? White rabbits means they are homozygous recessive, right? So the easiest way of breeding the white rabbits will be you take 2 white rabbits and make them breed. Right? Then the entire F1 will be white. All their offsprings will be white. Do you people agree? Quickly, can I have some responses? So we'll continue this session again for another 5 or 10 minutes, guys. So please be active, okay? And then we can talk about your personal questions or doubts for any question that we solve together, okay? Or we can solve some extra questions in the Zoom link. Okay, we have to stop the live session here. I'll share the Zoom link with you all, and we can do the rest of the questions in the Zoom class. Okay. Yes. So do you all follow so far? So the easiest way of growing breeding a white rabbit will be you cross 2 white rabbits. You breed 2 white rabbits. Correct? But that is not possible, or which is not economical. Why? Because the female white rabbits, they breed poorly. Okay. So that's why the farmer rejects this way. He doesn't go this way. And instead, he buys two black rabbits, okay, and breeds them. Okay. Now, when he bought these black rabbits and breed, the next generation, he got 6 black and 2 whites. All right. Now, let's write this cross here. So, this will be our parental cross. So, in the parental generation, what the farmer did, he took 2 black rabbits. Okay. Black means they both have at least one capital B allele. The other allele we don't know, so we are leaving it blank. Now, the F1 generation, what he has got? He got 6 black. Okay, the genotype again, we will write one capital B for sure. Other allele we don't know, leave it blank. And 2 whites. Now, this whites for sure, we know the genotype. It has to be small b, small b. Okay. Now, by looking at the genotype of these 2 whites, you will also understand the genotype of the parentals here. The white rabbits have two small b alleles. Okay. तो ये 2 small b alleles कहाँ से आए? One from each parent. Right? That means यहाँ पे अगर parental rabbits दोनों black है, तो दोनों का capital B allele तो show हो रहा है, but the other allele which is not showing is small b. So basically they both were heterozygotes. That is when it was possible for them to produce these white rabbits. Okay? So यहाँ से हमको ये parental जो farmer ने breed किया था उसका genotype पता चल गया. Okay? But this farmer did not definitely want to make more money, so he sold off these white rabbits. Okay. Now he want to breed more white rabbits. Okay. So the question is asking that if the farmer performed random crosses between the F1 rabbits. So यहाँ पे उसके पास क्या बचे? ये 6 black F1 rabbits बचे. Right? So अगर ये farmer इन 6 black rabbits में से कोई भी 2 rabbit को randomly उठाता है and mates them, what is the chance that in the F2 progeny he will get a white rabbit? Okay, now for this, let's do the cross here. So I will start with the parental cross. So parental cross, the first generation ka jo cross tha, it was this. Okay, now I will make the Punnett square. So this was the F1. Okay, this was white and the farmer sold it off. Whatever is left is black. Okay. Now, in the black rabbits, what do you have? We have either homozygous dominance or heterozygotes. Okay. Now, if you take homozygous dominant, okay, if you take 2 homozygous dominants, are you ever going to get a small b, small b? Not at all possible. Similarly, if you take one dominant and one heterozygote, then also the chance of getting small b is absolutely 0. Okay. So when can you get small b, small b? Small b you can only get if both the black rabbits that the farmer picks up from the F1 are heterozygote. This is the only possibility to get the white rabbits. Okay. So now the black rabbits which are left, so there are chance that they are homozygous dominants or heterozygotes. Okay. So randomly if he picks, what is the chance of heterozygote? So among the entire black progeny, okay, what do we have? What is the chance probability of heterozygote? So it is 2/3. Whereas probability of homozygous dominant is 1/3. Yeah, but Bioteki, remember, small b is not at all there. It was white rabbit and the farmer sold it off. Now he has only the black rabbits left. Okay. So now if he wants to grow white rabbit. Okay, so there are 3 events that needs to be taken care of. Okay. So what will be the probability of F2 white? Okay, so for that, no, I'll not write down probability. So for F2 white, okay, first thing is that the one F1 that is taken is heterozygote. Another event that is required is the other F1 that is taken is also heterozygote. Okay, and the third is that the cross of these 2 F1s produces homozygous recessive. So these are the 3 independent events which should occur simultaneously in order for the F2 to be white. So if we have to calculate the probability of F2 to be white, okay, so that will be given by, again here we have to apply the multiplication rule because these events are independent. So the probability of these events happening together will be multiplication of their individual probabilities. So probability of the first F1 is heterozygote multiplied with the probability that the other F1 is also heterozygote multiplied with the probability that their cross produces homozygous recessive. So what is the probability of F1 to be heterozygote? We have already found it out, 2 by 3. Multiplied with, again, 2 by 3. What is the probability that if 2 heterozygotes crosses, then you get homozygous recessive? What is the probability of that? 1 by 4. So what is the probability that F2 will be white if you cross 2 F1s? Will be equal to 1 by 9. Okay, yes, Afifa, the answer is 1 by 9, option C. Okay, guys. So how was it? I did not even notice the time flew so fast, and our 45-minute session is coming to an end now. So guys, we all from Biotechnica, we will be coming live, okay, every day, and we will discuss more questions. Okay, so you keep a watch on our YouTube channel, and it will be already advertised like which topic will come session we will do. Okay, so again, next day of the week also, like tomorrow also, there will be session. Every day there will be sessions, and I hope to see you all so that we can discuss more questions and we can solve more questions together. So I request the team to share the Zoom class link in the chat box so that the students who have personal questions, who have any personal doubt, one-on-one session we can have in Zoom. Okay, and definitely some free goodies are there for you. Team, please load it. Yes, more sessions will be coming by Techie. We are coming with more sessions, don't worry. Okay, yeah, Dev Malya. All right. So next time, please come on the session, right? So come on the session on time. Please take care that your networks are all fixed. There was some problem from my network also. I'm extremely sorry, and I will like from next session that won't be there. So team, please log in. Please give the Zoom class ID as well, so that the students can join. All the students, whoever have questions, please join the Zoom class link for one-on-one doubt clearing session. Okay, it's there in your chat box. Everybody in chat, our team has already posted. Please join the link. All right. Thank you, everyone. I hope to see you all on even more. If you have your friends, please let them know that we are doing this question discussions in the live sessions. Let them know, let them join. Okay. And we can solve more questions. All right. Thank you, everyone. Hope to see you all in the Zoom class. Yes, I'm just joining the Zoom link now. Thank you. Bye-bye.